So I work on myrtle rust. So myrtle rust is spreading around worldwide and is actually now in Singapore. It's a biotrophic fungus, and what biotrophic means, it actually needs a living plant host to survive. It infects many plants of the Myrtaceae family. There's over 2,000 species in this plant family. And it's really important because actually 10% of the Australian native flora are these Myrtaceae plants, such as eucalyptus trees, paperbark trees, and the leaf-pilly trees. And they're koalas, and many people love eucalyptus trees. It'll defoliate the plant, and it actually can kill the plant. And some plants are becoming endangered because of this fungus. So I devised a project with 4 aims. We really wanted to create reference transcriptomes for some of these plants and the fungus. We wanted to look at what transcripts are expressed in the plant in response to the fungus, what isoforms are being used, and what methylation patterns we can see in the plant. So we devised this experiment where we infected the plants and we had a mock control. It requires dark, and then we put it into the growth chamber. We performed nanopore direct RNA sequencing, which has many benefits, which I can go into at the break. And we really created high-quality transcriptomes for the plant and the fungus. So for the plant, we got 33,000 gene models, which is about what we expect. And we saw 124,000 different isoforms, which is really novel because actually the literature for many of these plants reports 0 isoforms at all. For the fungus, we got a really great transcriptome, which is over 7,000 gene models, and also many isoforms, about 14,000 isoforms. With these reads, we're able to do differential transcript expression. So really we're able to replace Illumina short-read RNA-seq methods, and we get better results because we're able to resolve all these isoforms. We detected thousands of transcripts that are increased in expression. Some of these, for example, are enzymes that are actually degrading chitin, which is actually a crucial component of fungal cell walls. And we're also seeing many transcripts that are decreased in expression. The interesting thing, what we found is actually 60% of our genes actually have an isoform. Most of the literature does not report any of these isoforms, and by looking at these isoforms, we're able to see isoform switching events. We saw these about 500 different isoform switching events, 300 in early stages, and then we see as the infection progresses across the plant, we see even more isoform switching events. And what do I mean by isoform switching events? So before the infection, we might have this isoform X, and as the infection comes along, we actually see a decrease in this isoform X, and we see a transition towards a preferential usage of isoform Y. As the infection progresses, we actually see a switch again. Isoform Y becomes decreased in expression, and we see a transition towards preferential usage of this isoform Z. And as you can see, it's the 3' end of this isoform is changing quite drastically, which actually can change the function of the protein or the localization of the protein of that gene. What we've been looking at into now is actually what methylation is occurring during this infection process, and what we see is actually there's a lot of methylation being written. So this is dynamic methyltransferases adding methylation to the RNA. We also see dynamic removal of the RNA as the infection progresses from the plant RNases. And also there are actually RNA binding proteins that will recognize these methyl marks on the transcript and associate with that RNA. So actually we're investigating this process because we have a dynamic mixture of RNA methyltransferases and erasers from the plant and from the fungus happening in the system. Thank you for listening.